Welcome, everybody. Welcome to our second department seminar this semester. Today, it is my pleasure to introduce Dr. Brian Oakley. So Brian Oakley is a professor specializing in microbial ecology and bioinformatics at Western University of Health Sciences College of Veterinary Medicine. I had the privilege of meeting Brian back in 2019 when I was coming back from my second postdoc at the University of Rhode Island and wanting to settle in California for whatever reasons. I walked up to this man's office and I knocked on the door and I introduced myself and asked for a job.
Basically.
And we've been good friends ever since, actually. I think we've been hanging out, having a good time. All right, let's see. I have some notes. Brian earned a Bachelor's of Arts at the University of Vermont, and I know I didn't find what that bachelor's was in, but it's something interesting. Like
East Asian Studies
East Asian studies,
and political science.
It's a Renaissance man right before us, Okay, and then you went on to study Japanese, right? Formally,
Correct.
At UC Berkeley.
Correct.
Yeah. So the Japanese language, right?
Yeah.
I should mention he's a published author in the Japanese language. He's got a couple of books, children's books.
What I translated one book from Japanese to English, a children's book. So
Anyway,
mostly pictures.
that's his most impressive achievement to my son, Augustus.
Okay.
He still knows you as the guy who writes Japanese children's
That
books.
was my first publication.
That's right, first publication. Then Brian went on to earn a master's at UW, University of Washington. You have an emphasis, that's right, it's rocking UW.
Represent
Do you have an emphasis for your master's, sir? That was ecology, right?
plant ecology,
Plant ecology, so
botany and plant ecology.
right, and like something to do with forest fires, correct?
That's correct.
Yeah, after that, Brian stayed at UW and did a PhD,
Yep.
same team too. From there, I guess continuing his tenure at UW, Brian went on to do postdoctoral work there. And eventually moved on to the Fred Hutchinson Center, right, for Cancer Center.
Yep.
That's also huge in genomics, right?
Yep.
Guessing that's when you started to embrace the bioinformatics part of it all. At some point after that, or some point short after that, Brian went on to receive the prestigious Marie Curie Fellowship.
Well, I
And
was
went
part
on
of
to-
a project. I wasn't personally to me, but
Okay, as
Right.
part of a project,
Yeah.
Brian became a Marie Curie Fellow.
not quite. Okay.
Okay, something like that.
I'll take it.
Butchering it. All right. Anyway, he did this work at the University of Warwick in the UK.
That's correct.
Okay, his first job after that was at the USDA ARS, so that is Agricultural Research Center
Service.
Thank you. And he was there for 4 years. And there, I mean, it's an entire body of work on the microbiome of food animals, which is, I guess, the beginning of like a lot of the stuff that we're going to get to hear about today. From there on, Brian went on to accept a professorship at Western University, College of Veterinary Medicine. That was 2014, and that's where he is currently. I should mention, Brian is the most highly cited author at the university.
Yeah.
By far,
Sure.
yeah. So it's been an extreme privilege to be around him and his ideas, and obviously a profoundly influential scientist in my life, and a really good friend that I esteem dearly. So anyway, with all of that said, here is Professor Brian Oakley.
Thank you. That's so nice. Thank you, Gustavo. Appreciate it. Yeah, that's all true, mostly true. The fellowship wasn't to me. I was part of a larger project at the University of Warwick in the UK. But I was joking and I would give you unsolicited career advice. My career has kind of taken me through many circuitous twists and turns. But I've tried to always do things that are interesting to me, and follow opportunities that were presented to me. So I've worked on a lot of stuff in my career. I mean, basically, as Gustavo was saying, sort of started out more as an environmental microbiologist, and then kind of got into the project I worked on at the Fred Hutch Cancer Research Center was part of the human microbiome project. And then basically kind of develop those skill sets. And as you said, like bioinformatics and so on was becoming more and more, common and widely used. And then so a lot of the kind of questions and tools and techniques that I had in my toolbox became applicable to like animal associated microbiomes. So that's what I want to talk to you about today. I've been at Western for 10 years. It's just down the road, as you guys know, from Pomona. Just hopped on the train this morning. Love the train. Just got off and there was Gustavo waiting for me. So more than happy to be here. I was here a year ago or so for the Gene Lab thing that you guys did, which was great. So yeah, this is kind of it is my title today, is managing the microbiomes of food animals to improve sustainability. So I'm going to kind of walk you through sort of the rationale and the reasoning behind that and give you some like specific examples from my own research about ways in which we can do this. So there's a background about Western. If you guys are ever in Pomona, like feel free to shoot me an email. Happy to show anybody around class or on campus. I teach, so our students there, I'm in the College of Veterinary Medicine, so our students they're getting their DVM, Doctors of Veterinary Medicine, so it's a 4-year program. Most of our students, almost all of them, want to be like practicing clinicians. They want to be clinical veterinarians, like mostly working with small animals in like a suburban animal hospital, whatever. Not the majority of our students are not interested in like food animal medicine per se. But kind of the little hook that I use to get them interested in the courses that I teach is I say like, hey, all animals are mostly microbial. These are true statements. Okay. So a lot of the data that we know about microbes associated with animals is coming from the human side, and it's kind of sort of transferred over to the veterinary side. So these are all true statements and illustrated in kind of these fun ways. So on the left is the number of microbial cells relative to the number of human cells in and on our bodies. And then on the right on this panel here is a number of genes. Okay, so whether you look at it by like cell count or gene count, all animals are mostly microbial. 50 to 90% bacterial by cell count and 90 to 99% bacterial by gene count. Okay, so really, like you could think of ourselves and all of our companion animals and food animals as really, we have a perceptual bias that I see like Oscar is this human-sized shaped individual in front of me, but really what I'm looking at is like a bag of microbes. Okay, it's a collection of microbes, all right? They're not just like incidental hitchhikers hanging out. They're doing like really, really important functions. And so that's kind of what we're going to talk about today. And from a food animal perspective, there's a lot of ways that we can kind of, if you think of like knobs and levers and dials or, an app or whatever, that we can kind of optimize these communities associated with our food animals in ways that might be helpful to us, as whether we're food animal producers or we're consumers. We're trying not to get sick from consuming different food animals, et cetera. Okay. So let's talk for a minute about this term microbiome. So we've mentioned it a couple times. And it's a term that has kind of, I think, trickled over from being like in the scientific literature to now you see it all the time, right? You see like, lotion ads and they say like, hey, enhances a healthy skin microbiome or whatever, okay? I think this is actually a pretty good term. If you think about the term biome, right, think back to, high school biology or whatever, right? It's like the biome of like the coral reef biome, the tropical forest biome, et cetera. So, right, these kind of large scale ecosystems. And I did this experiment, just kind of back of the envelope stuff for fun a few years ago, where if you take like the surface area of a typical biome, so I just Googled the Masai Mara, the Serengeti ecosystem, apparently it's like 25,000 square kilometers in size. And I just for fun, I said, okay, what's the scale of like an individual animal that might inhabit that biome? A zebra, if we say a zebra is like 2 meters, say 6 feet in length, something like that. Okay, just obviously a rough estimate. All right. And then if we look at like, okay, what's the surface area of the human, of like our GI tract, the surface area, if you were to like spread out our whole GI tract, it's about 40 square meters, okay? And a bacterium, you could say a typical size of a bacterium might be 2 micrometers, okay? So if you take the ratio of the size of the individual here to the size of the biome in which it's living, the ratios are roughly the same. Okay. So you can think about an individual E. coli cell or whatever within our GI tract or the GI tract of your dog or cat or whatever. It's roughly the same, okay, physically, just the relative scale of it, as a zebra, say, for example, wandering around this big biome. Okay. So I think it actually is a pretty good term, like this term microbiome, because it really is like a biome, these kind of large scale things we think about, just like shrunken down everything about it. Okay. My example there, the Masai Mara, might not be the best example because it's relatively species poor And as we're going to talk about, most microbial communities, most microbiomes are more like a tropical forest where they're very species rich. Okay. So we typically have, like in our GI tract, all of us have like hundreds to thousands of different bacterial species. And then within a single gram of our like intestinal contents, we could have easily, depending on kind of the location through your GI tract, you could have like 10 to the ninth cells, so a billion cells per gram. Okay, so roughly like as many bacterial cells within a gram of your intestinal contents as there are humans on this planet. Okay, so kind of like mind-blowing some of these numbers. Okay. Helpful, I just include this slide, helpful to kind of think about a lot of diseases as kind of like they're a dysbiosis. Okay, so it's like a shift in that whole complex community. Okay, so if you just like as a simplistic model, like take this kind of healthy, the forest ecosystem, and versus this where it's like burned and everything is kind of, not clear cut, but burned across. A lot of human and animal diseases, I don't know if you can see at the bottom there, but like IBD, colitis, Crohn's disease, ruminal acidosis in cows. I've got a gruesome picture I'll show you in just a second. Necrotic enteritis in chickens, bacterial vaginosis in humans. I worked on that at the Fred Hutch. Periodontitis, foot rot, et cetera. All these diseases are basically like microbial dysbioses. Okay, so it's not like a single etiologic agent. It's not like, oh, you've got a salmonella infection or you have a Clostridium infection or whatever. It's like the whole community is like gone out of whack. Okay, so thinking about sort of these microbiomes, these microbial communities as like whole systems is really important. And it's a good kind of mental model a bit to think, okay, here comes my gruesome picture of a bunch of dead calves. Okay. This is out by the Ontario airport. There's still a bunch of dairy farms that are out there. Sometimes you can, if you guys can smell it here, but sometimes in Pomona, depending on the wind, we can kind of smell it. Long story, we won't go into details. It's actually fascinating. Ruminal acidosis, because of improper feed, happens all the time in both dairy and beef cattle. They're trying to get these animals to grow as quickly as possible. These animals are ruminants and they're used to eating like complex like forage, grasses, and so on. And we give them relatively simple grains. It's almost like a sugar high. These things are easily digested. When they're digested, they lower the pH, it becomes acidic, and that further shifts the microbial community. And then it leads to a whole bunch of kind of follow-on problems, bunch of physiological problems. And these animals end up basically, they get, one of the first symptoms, they get laminitis. So they get a problem with their hoofs. And they do that. There's a characteristic stance, it's called a sawhorse stance, or that they kind of go like this. And you can see, and then they're like, their feet hurt, so their hooves hurt, so they're walking tenderly. And then they start to get, their immune systems are compromised, so they get they're susceptible to airborne pathogens, so they get all kind of lung problems. So they have problems breathing. So you see them, they're doing this like kind of like gasping motions as they're trying to breathe. So I went out with some colleagues I work with at Western who are veterinarians, and we had to actually euthanize some of these animals because it's hard to recover once they get past a certain point. And then our students do like, this is called a field necropsy. Okay, so I don't know if this is like selling vet school to you or not, but so our students are like, oh yeah, look at all the pathologies in the lungs, and they understand everything that's going on. But for me, when I see that, basically the point of the slide I'm trying to convey to you is this is just a dysbiosis. Okay, this example of the whole community has gone out of whack. In this case, it's all because it all starts here because of improper feed. Okay. So we're going to talk about the importance of like feed and diet for kind of maintaining and nurturing an appropriate microbiome. But I just want to give you a little bit of background on kind of what Gustavo and I, one of the many things we have in common is we're both microbial ecologists. And so microbial ecology is looking at like who's there in a community, how many of each, like what's their relative proportions, and then what are they doing, like how all the bugs function, how do they contribute to the health of that ecosystem, whether it's like a deep sea ecosystem or an animal or us as people. And the reason that we can now ask all these questions is basically the reason that we all carry around these amazing like smartphones is all like technological improvements. Okay, so the cost of DNA sequencing in a nutshell has decreased. It's decreased even faster than we thought it would. Okay, so this little graph is from 2001 to 2022. This line here is Moore's law, which is like for computers. So Gordon Moore is one of the co-founders of Intel, the computer chip company. And he made this like prediction back in the day that I guess when you're like one of the original guys in Silicon Valley, it gets named after you and called a law. But and it was just like an idea that he had like a rule of thumb. Okay, that the speed of computers would double every 18 months, and the price of for that same amount of processing power would fall in half. Okay, so that was like Moore's law. Okay, so that's the prediction, and then this is the actual, this green line here shows what's actually happened with like sequencing, with DNA sequencing. And so you can see, and the scale over there is a log scale. Okay, so that's 100 million, that's 10 million, that's a million, that's 100,000. Okay, so you can see basically like it used to be prohibitive and basically impossible to sequence like the microbiome or our own genomes or whatever. Okay, you can look up the original cost of the Human Genome Project was like $3 billion, I think, or maybe, yeah, I think it was billion. And now you could get your genome sequenced if you're concerned about, prostate cancer risk or breast cancer risk or whatever. I think you can get your own genome sequence for $600 or $700 or something like that. Okay, so yeah, going from a few $1 billion to a few $100 is like transformative, right? Like, the cell phones that we all have now are like more powerful than the computers that they had for the Apollo space system. So point is we can ask and answer a lot of questions that we just simply didn't used to be able to because of kind of technological limitations. Okay, so we're happy to be alive right now and happy to be in this field, so that's a good thing. I just want to give you briefly, I don't know kind of your exposure and background to this, but so I just have a couple slides there's kind of two main flavors or types or tools of DNA sequencing that we do in microbial ecology. The first is sequencing the 16S ribosome RNA gene. Have you guys talked about this, like roughly? Okay, so it's this taxonomic marker gene, right? So we can take any sample. You can take a swab of your skin, you can run it through a DNA kit in Dr. Ramirez's lab. Super easy, just cookbook. We could show you how to run through the kit if you haven't done it. And within an hour, you can have basically a DNA sample that represents it's a purified DNA from all the bugs that might happen to be in that little patch of skin that you swabbed. Okay, you can send it to a lab. They do this 16S sequencing, and the beauty of it is, one of the many kind of the most powerful aspect of it is that you don't need any like prior knowledge. You don't need like a specific PCR primer set for E. coli or whatever. You don't need to know what's there. You just do this like broad range sequencing of that sample, okay? And you get back data that is basically, you end up with data like this. It's like a census, if you just think, okay? So you're just doing this like unsupervised, unclassified census. I have no prior knowledge of, of the composition of this community, but I can send a sample off to a lab. And again, back to my previous slide, for relatively cheap, for like a few dozens of dollars or something per sample, I can get a census, and then I can compare. I can say, okay, what happens when I'm going to have Oscar's diet switch to a vegan diet, and I want to follow what happens in his GI community every day for the next 2 weeks. Okay, so I could do that, and I could say, okay, here's Oscar on day one, and then I can go through, and then I can compare and say, whoa, look, there's a shift in all these beneficial bugs or whatever I might find. Okay. So that's 16S RNA gene sequencing. RNA is the ribosome RNA gene. And then again, it's just this taxonomic marker. So it tells you like who's there. So it's just like a taxonomic identity of the bug, but it doesn't tell you what they're doing. And that's actually a really important distinction because, for example, like not all E. coli are not identical, right? There's like benign E. coli, there's pathogenic E. coli. So for me, just to say that, oh my gosh, you have E. coli, like, right, there's a follow-up question is it a good one or a bad one, right? So you kind of need to know more than just like the name of the bug that's there. Okay, so to do that, to answer those kinds of questions, we use this technique called metagenomics, which is very similar, except there's no like, we're not looking at just an individual gene. We're not looking just at this 16S ribosome RNA gene as this taxonomic marker. We're looking at all the DNA that's there. So I'm gonna do the same thing I did before. I'm gonna take a swab from my skin. I'm going to run it through this DNA prep kit. I'm going to get in my tube a little purified batch of all the DNA from all the bugs that are there. And because of my slide about how cheap DNA sequencing is, now it's so cheap, I don't need to just focus on a single gene. I'm going to sequence all the genes from all the bugs that are in that sample. Okay. And so that's why it's called, this is also a good term, it's called metagenomics. So genomics is like sequencing the genome of an individual organism, whether it's you as a person or E. coli strain that you've grown in the lab. Okay. In this case, we're sequencing the full genomes of all the bugs that are in that mixed sample. And so it's called metagenomics. Makes sense, right? Okay. And if you think about all the types of information you can get from that, this is just this kind of a schematic illustration. Like here's our complex community, right? All these different kinds of bugs. And we're just gonna like, again, sequence everything that's there. And then we're gonna get not just like a taxonomic identity, like, oh, we have 10 E. coli, we have 5 salmonella, we have 1 clostridium. We're gonna get like, hey, these are all the genes that are there. So, oh my gosh, look, you've got a bunch of antibiotic resistance genes. You've got a bunch of genes involved in carbohydrate metabolism. You've got a bunch of, you know, genes involved in protein metabolism, et cetera, et cetera. Okay. So those are really the 2 main tools that we use. And I wanted to show you those just to be sure we're on the same page, because the slides I'm going to show you coming up about my own research are basically all just applications of these 2 techniques. Okay. So we have 16S sequencing and metagenomics. Okay. All right. So going back to this idea of like all animals are mostly microbial, and it's not just like they're not incidental hitchhikers, they actually do really important things. Okay. I hope I'm not that blunt. Okay. There was this landmark experiment way back in 2006. This guy was a postdoc at, I think, Washington University in St. Louis. He did this experiment, kind of blew everyone's minds, and he got a faculty job at Harvard. And that's kind of what you dream of, and then that never actually happens. Okay. All right. Full disclosure, I think he left Harvard because it was a really difficult environment. He went somewhere else. Okay. Anyway, he did this super cool experiment where they took GI material from twins, and one twin was obese, the other one was lean, and they gave it to mice that were genetically identical mice, and they fed them the same diet. And lo and behold, depending on the phenotype of the donor, that was reflected in the phenotype of the recipient mouse at the end. Okay, so the mice that received this fecal material from obese twins, from the obese donor, they became obese. The mice that received the fecal material from a lean twin, they stayed lean, even though they were the same genetics and the same food. Okay. And people are like, well, like, so the microbiome, the gut microbiome, controls this key phenotype. Okay. So you can imagine from the human side, this is before Ozempic and all this stuff, right? Okay, so from the human side, people are like, hey, can we make like, you know, the skinny pill probiotic or whatever, and I can still eat my 5 guys and fries, I don't have to ride my bike all the time, whatever. Okay, from the food animal side, we're kind of interested in the opposite side of that equation, right? About 70% of the cost of like a broiler chicken or whatever, a chicken that you buy at Trader Joe's or whatever, is the cost of the feed. Okay, so the cost of the feed is like the most, has the most influence on the cost. It's the biggest cost for the producer. Okay, so if you're a food animal producer, if I grow chickens, if I can feed my chickens a little bit less, and remember, I probably got like tens of thousands of chickens, so it can just be a little bit less, but multiply across that many birds, right? Then, but they still reach the same body mass, then it's gonna translate to a lot of money for me. So I'm really interested in that. Okay, all right. So that this experiment really set the stage for a lot of subsequent work, okay, including a lot of our own work. And so Gustavo and I have done similar experiments of this with chickens, and I'll describe that to you in a second. But if you think about it, really, this is still pretty black box. Like nobody knew why. They just observed like, hey, we have this outcome here. This is the phenotype. What's the mechanism behind it? Which bugs in particular are responsible? Like at that point, 2006, nobody knew. Okay, so that's what we're trying to figure out. Okay, this is a busy slide, but this is super cool. If you guys want to ever look up this paper, I don't know if you're going to share these slides or what, but this is an amazing paper, my all-time favorite papers from 2016, where, and again, this is just showing the importance of our gut microbial communities for our health. Okay, and this is again true for us and our companion animals and our food animals. Basically, what this guy did is their lab, they took a group of mice. They fed half of them a fiber-rich diet, as I've kind of illustrated here, and half of the mice they fed a fiber-free diet, as I kind of, this is like my 16-year-old son's diet here. That's kind of my joke about that. All right. And long story short, what they found was that in the mice that were fed the fiber-rich diet, lo and behold, when they looked at what happened to their gut microbial communities, their gut microbiome, they had a bunch of all these different species of fiber degrading bugs. Okay, so many, like many different species. And that led to, they had the mice had a really thick mucus layer, okay, in their GI tract, which is a really good thing, because if you think about our GI tracts are basically like this alimentary canal of the outside world going through the middle of our body, and we don't want sepsis and et cetera, right? So we want to keep the bad guys out and keep them separate. And one of the key ways that we do that is this mucus layer through our GI tract, okay? So the mice that were fed, again, this fiber-rich diet had this really healthy gut community, really healthy, like kind of, you know, physiological parameters of their gut. In contrast, the mice that were fed this fiber-free diet had a proliferation of this little red color here. These are the mucus degrading bugs. So the microbes that became really abundant in that group of mice were actually eating the mucus layer of the mice, okay? And so those mice had a really thin mucus layer, all right? I saw the author of this paper present this, and he's like, oh my God, like the communities were totally different. We could see these physiological parameters were different, but he goes, the mice were still, they were like performance, so phenotype wise, they were basically equivalent. And so we were kind of like, oh, that's too bad. Until they challenged those mice with this yellow thing here. This is in their cartoon, is a mucosal pathogen. So it's closely related to E. coli. Okay, when they infected these mice, they challenged them, it's called, and they challenged them with this mucosal pathogen. Like this is what happened. So this is the weight change in percent, is 100%. This is days post-infection, so going from 0 to 10. And this group here is the mice that were fed the fiber-free diet. Okay, so these are the mice with the crappy, if you will, the bad gut microbiome that have this really thin mucus layer. And lo and behold, when they got sick, when they challenged them with this mucosal pathogen, they had to actually euthanize. They stopped the experiment because these mice, they lost like 30% of their body weight in 10 days. So they were like, the veterinarian in charge of the study is like, yeah, this is no longer humane to keep running the study, you have to stop. So they'd euthanize all these mice. Okay, so I teach a food safety class at Western, and this is one of the papers that we read. And it really, the take-home message is like, we as people need to eat a healthy diet, because that gives us a healthy gut microbiome, which does many things for us. But one of the most important things is it protects us against foodborne pathogens. Okay, this is like really nice, really bulletproof data that really makes that point. So I think that's really nice. Okay, let's talk just briefly about what we mean by a fiber-rich diet and what is fiber, because I think there's a lot of kind of popular misunderstanding, and we see all these now again, like it's all becoming our kind of popular lexicon about like, hey, a prebiotic soda and whatever, probiotic this, probiotic chewing gum or whatever. Okay, all right. Fiber. And this links back to the diversity of microbes. Remember, I said there's like hundreds of species, okay? It turns out through evolutionary time, as Gustavo often talks about, is we have had this amazing like ecological diversification where different bugs specialize in digesting different fiber sources, okay? And that's one of the main things that they do for us, okay? So we as humans, we've like outsourced our metabolism. We don't have the genes, we don't have the ability to digest a lot of the things that we eat. We rely on our bugs to do it, our gut bugs to do it for us, okay? What this graph shows is a complex, but basically every bar here is a different enzyme that's encoded by a microbe it's an enzymatic scissors to break apart a different fiber molecule. So fiber is actually like a whole world of complex carbohydrate chemistry, okay? So, and we can see that a little bit. So the colors here, so these are the kind of the families of fiber types. So this is cellulose and other beta glucans, hemicellulose. So these are like part of the plant cell walls, right? Like the bran or the wheat or whatever. Pectins, starch and storage glycans, fungal cell wall mannins, et cetera, et cetera. And again, every bar here is a microbial enzyme that's encoding an enzyme that breaks apart one of those particular carbohydrate molecules, okay? So we have beta galactase, beta xylanase, alpha L rhamnase, okay, whatever. So I'm not an expert in carbohydrate chemistry. These people spend their whole life studying that, but carbohydrates are these long, complex molecules with all these different linkages, remember back from chemistry, and all those require different enzymatic scissors to cut all those up, okay? And what does it, again, is not the host animal, it's the microbes. And because the world of carbohydrate chemistry is so complex, like all the plants and so on that we eat, right? That's why we need this like diverse microbiome to be healthy, because we rely on it to digest all these things. Okay. So that's the point of that. And again, this is true not just for us, but for all animals. And there's some really extreme examples. Okay, so here this is kind of the take-home message is carbohydrate digestion has been outsourced to GI microbes. Okay, so again, that's one of the most important things that they do for us. And this is they sequenced the giant panda. They did a metagenome sequencing. So again, they're just taking the DNA of all the bugs that are in a fecal sample, whatever, from a panda, and they're sequencing all those genes, okay, of all the bugs that are there. So it's a metagenome, right? And look at what they find. These are all basically like carbohydrate digestion enzymes involved in that. So beta glucosidase, beta galactosidase, beta manosidase, blah, 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 blah. List goes on forever. Okay. So pandas are there, they're chewing on their bamboo, whatever they do, right? They don't have the enzyme, they can't digest that themselves. Bamboo is like pretty recalcitrant. It's not like a cheeseburger or something, right? It's like chewing on bark or whatever. Okay. So these are all enzymes that are encoded by their gut bugs. They're absolutely essential for them to be able to subsist on that diet. Okay. So that's one really important point. Okay, so let's come back to kind of this idea, right? That like microbes can confer a particular phenotype and have, really maybe with like from a food animal perspective, maybe like really like economically important implications. And let's link it in. So Gustavo said, like, I kind of started my career like doing more sort of like plant ecology and thinking about like ecological ideas. There's a few ecological ideas here that are really important that tie in really well. So one is like the importance of early life influences. So this is a picture of a dairy calf being fed colostrum. So colostrum is like the first mammary secretions after birth, right? And so it's really high in milk fat. In ruminants, it also transfers a bunch of immunoglobulins, has a bunch of kind of immunity, passive transfer of immunity aspects. But the main thing it's doing is providing a bunch of oligosaccharides and a bunch of milk fat to the calf. So the idea that like early life influences are really key has been shown over and over and over. Okay. Other examples of that are it's been shown that human infants that are born vaginally versus C-section have different skin microbiomes, different gut microbiomes. Human infants that are breastfed versus formula fed have different GI microbiomes, at least for a while. That I think they kind of end up converging back. So the idea that like early life influences, sometimes people talk about like successional trajectories. Okay. So if you think about a trajectory, like a little difference in angle at the beginning kind of amplifies out, right, to a big difference in absolute magnitude at the end. Okay. So with food animals, what we're talking about is like, can we do a little bit of nudge at the beginning, like with the chick, in the case of a chicken, when the chick first hatches, and then could we end up having that have a significant effect on the phenotype at the end of the life of the bird? Okay, so I'm going to show you some examples of how we do that in a second. Okay, so we'll leave it there. So, all right, so why, now I wanted to just give you a couple slides on why, like the rationale of why we should kind of use these tools of microbiome science and try to improve the microbiome related to food animals. So I already talked about like the cost of producing and can we improve like what's called feed efficiency. There are multiple problems with our current system of of protein production. Okay. Greenhouse gases. Okay. So food animal production contributes a lot of greenhouse gases to the atmosphere. We use a lot of antibiotics to produce our food. And we also still have a lot of people getting sick from foodborne illnesses. Okay. Everybody okay? Okay. All right. So I just want to walk through these issues really quickly. This is okay, kind of like one slide per. All right. Greenhouse gas emissions from food production. It basically accounting for about a quarter of greenhouse gas emissions are produced via our food production systems. Okay. Cattle and beef production in particular is really bad. Cattle production is really feed inefficient. You've learned a little bit, I hope, about like methane, methanogens in ruminants are producing methane, which is a powerful greenhouse gas. Okay, so we're producing a significant amount of global greenhouse gases from the current ways in which we produce and eat food. Antibiotics, so I don't know how much people know about this, but antibiotics are used, still commonly used in food animal husbandry, not to treat disease. So it's not like a therapeutic use. It's called a subtherapeutic use, or like a prophylactic use. Because in the 1950s or something, people were like, hey, if we give our cows and chickens, if we give them like low levels of antibiotics, they become more feed efficient. And again, we can like, if I can feed my food animals less feed and I produce the same amount of biomass, then I make more money. So let's just feed them antibiotics and we can make more money. And that's good. Okay. This has become, we have since recognized since the 1950s, this is a significant public health problem. Okay. So this is just a little chart to show that roughly about 2/3 of all antibiotics produced. Okay. So this is showing that 65% of all antibiotics produced are used in food animal production. Okay. And about 35% are given to humans. All right. And then this is a breakdown by species. So about 25% given to cattle, 26% to pigs, and the remainder to chickens and turkeys and so on. Okay, so we're using a lot of antibiotics prophylactically, like subtherapeutically in food animal production. Why is that bad? We could teach a whole class, but basically it's a perfect system to select for antibiotic resistance. Okay, so a lot of the antibiotic resistance genes have existed for forever. So bacteria are engaged in chemical warfare with each other in the soil is kind of the classic example. And you can go to like Arctic permafrost that are kind of, without free of human influence, and you can find antibiotic resistance genes. So we haven't like created these genes like de novo from scratch just by using antibiotics. What we've done is we've just select for them, right? And then a lot of these things end up with they're leading to hospital acquired infections, and people now have infections where antibiotics are not working anymore, right? So we've heard about these problems. Okay. So this slide is basically making the same point. So here's medically important antibiotics over here. So you can see like and this is use in food animals, okay? And then this is through different years from 2012 to 2021. And so you can see like tetracyclines are used really heavily in food animal production. There are some regulations in 2016, I think, that have decreased it, but still obviously it's decreased it, but it's nowhere near zero. And we're still using a bunch of what are considered non-medically important antibiotics. Okay, so this is a problem. I'm just making the case that antibiotic usage and overusage is a problem of food animal production, and it's something that we can solve with our toolkits of microbiome science. Okay, and I'm going to give you specific examples. Okay, the last kind of problem about food animal production that, again, I think we can solve with a lot of kind of our microbiome optimization ideas is we still have a lot of foodborne infections. Okay, so these are like some of the more common bacteria that are associated with our different foods. So Campylobacter is kind of typically associated with chickens. Campylobacterosis is one of the most common foodborne illnesses people get. It can lead in some cases to a follow-on disease called Guillain-Barré syndrome. It's like paralysis. And typically people recover, but you can be like in a coma for a few weeks or something like that. And I think some people have like kind of debilitating paralysis that lasts for a long time. Salmonella, we've all heard about. Shigella, S-tech E. coli, these Shiga toxin producing E. coli, Cryptosporidium, Vibrio, Listeria. There's always like, in my news feed, it's probably biased to me, but there's always like outbreaks of Listeria in cheeses and stuff like that. And Listeria is not super common, but it's really deadly. And particularly pregnant women are particularly susceptible. So Listeria is a particularly nasty pathogen. Okay, so I've just given you kind of a really brief overview of why kind of problems with food animal production. And again, this is kind of our central question. Can microbiome science, just speaking broadly, can the tools of microbial ecology, can they be applied to sort of solve or improve some of these problems? Okay. All right, so the short answer is yes. And I want to give you 3 examples from my own research, and then that'll be it. Okay. So 3 examples. I want to show you how we've figured out how we can suppress pathogens. We can identify alternative antimicrobials. So if we want to control microbial populations, we don't have to use these like broad spectrum antibiotics. We can be a little bit more targeted. And then the last one is something that Gustavo figured out in my lab, which is how can we modulate the hydrogen economy, which is a super fascinating story. Okay. All right, so the first story I want to tell you is when I started at USDA, it was what's now called the National Poultry Center. And before DNA sequencing tools were available, it was people would do all these studies, but they really had no technical ability to do any of this kind of sequencing stuff, and they couldn't tell like what bugs were in a community, but they could feed them to chickens and they could see like, hey, is this working? Is this producing a desired outcome? Okay. So way back in the day, this is from 1995, a bunch of guys who worked there before I did, they made this thing called a mucosal competitive exclusion culture. And what they were doing, just in brief, was they were taking like gut scrapings from chickens and they would put them into vials in the lab, give them some growth media, kind of transfer it until they thought it was stable. They would feed it back to chickens and then they would challenge those chickens. They try to infect those chickens with salmonella and campylobacter. And they would say, can our culture, will it competitively exclude, will it reduce the colonization of the pathogen? Okay, so competitive exclusion, that idea has been around for a long time. It's super simple. If you think of just like niche exclusion, that like if the ecological niches in your gut ecosystem are like the chairs in this room, you want to fill the chairs with like all your friends. And then when your pathogens come in, there's nowhere for them to sit. Okay, so that's kind of the idea. So they were like, could we make like a beneficial culture, feed it to chickens, and like have it have this protective effect where the pathogens come in and there's nowhere for them to land? Okay, but again, they didn't have any tools to be able to see what was in these vials. They did it empirically. So they did this experiment like dozens and dozens of times. Like, I don't know, try this one, make another batch, try this one, make another batch, try this one. Hey, it looks like batch 3 works the best. Okay. So they found like batch whatever 3 was the best one, and they got a patent for it, and they tried to market it, and they were like, this, you can't sell this. The FDA said, you can't sell this stuff because you don't know what's in it. It's a product of unknown formulation or something. It's not like it's not like a, Tylenol where it's just Tylenol. It's like a whole mixture of all the stuff that they didn't really know what it was. Okay. So that was kind of the end of that story. And they had this patent and they thought they were going to get rich and famous and they got their government pensions. And I guess that was the happy ending for that. Okay. I came along like years later and this stuff was literally sitting in a freezer. And I thought, I've just been doing all this 16S sequencing and now we've got all these bioinformatic tools. Maybe I can pull these old vials out of the freezer and we can see what was in it. And maybe we could even like start to understand like how it actually worked. Because they showed it to be efficacious. That's why they got a patent. It actually worked. It reduced the colonization of salmonella and campylobacter. It's just they didn't know what was in it and they didn't know what it did. Okay. So I sequenced it. These are like replicate vials. So I just pulled a bunch of vials out of the freezer. We could see what was in it. I don't need to go into details, but so this is a list of the bugs. So this is 16S sequencing. So this is just a census again, right? So it just tells me like who's in there. All right. So that was helpful information that wasn't known. We can do metagenomics again, and then we can, so what I decided to do is like compare it to like a normal healthy set of birds. Okay. So we can see how is this like, it's kind of a derived culture because they took it out and they cultured it in the lab and so on. How is it different from like what you normally find in a normal chicken? It's kind of the question we're asking. Okay. So this color here, this our MCE is the mucosal competitive exclusion. Those are the secret old vials that were in the freezer. And we can compare it to, these are just like regular chickens, okay? And long story short, we don't need to look at these in detail, but the take-home message here is we could see just that the community that was in those vials, the gene repertoire there was different than a normal kind of a chicken microbiome. Okay, so then the question is like, how is it different? Like what's actually different? And because we knew that it was efficacious, maybe then we could start to understand like its mode of action, like how is it working? Okay, so now we're doing metagenomics, so we can say, okay, so what specific genes are like enriched in these old magic vials compared to a normal chicken microbiome? Okay, and so now we can drill down and we can see, okay, here's like 5, in this case 5, there's 5 sets of genes, 5 metabolic pathways that were significantly enriched. I just wanted to highlight a couple. So this one, I know you can't read it. This one is glycosphingolipid biosynthesis, and this one is secondary bile acid biosynthesis, okay? And they're enriched. In this case, here's our mucosal competitive exclusion, this red bar here. Okay, so those pathways were significantly enriched in these old magic vials, and maybe that's how they were working. Okay, so let's just take a quick look at these. So the first pathway that was enriched was this glycosphingolipids. So what are glycosphingolipids? So I did some research. So they're basically like glycoproteins that are expressed on host cell surfaces that are like binding motifs for pathogens. Okay, and it's been shown that if you add like exogenous glycosphingolipids, as this kind of panel is supposed to show, that so here they are here's our exogenous glycoproteins, these glycosphingolipids, they can bind with the binding site and they block the pathogen from binding to the epithelial tissue, okay? So that's super cool. So maybe that's one way, that's like a specific mechanism of action, right? That these old magic vials, that's how this stuff was working. Okay. I won't go into details here, but basically they might also, these glycosphingolipids might also have like some immune stimulation effect. So they kind of stimulate like NK T cells, natural killer T cells. All right, so that's one other possibility. So one of the other pathways we saw, there's 5 or 6 that we saw here. I'm just highlighting these 2. And the last one I want to show you is just a secondary bile acid biosynthesis. So what does that tell us about maybe how this stuff was working? Okay, so secondary bile acids. So bile acids are produced by the host, and it's been known for a long time that they're one of the main functions that gut microbes do. One of the things that they do is particularly Clostridia, and we could see in our census from our 16S data, there's a lot of Clostridia in those vials. Okay, Clostridia are well known to produce to convert primary bile acids, so cholate is an example, to secondary bile acids, and deoxycholate is a really good example of that. And we can find some papers to show that when you add deoxycholate, and here's like increasing concentrations of deoxycholate, that it decreases, there's a direct inhibition of salmonella ability to invade the host cells, okay? So this is probably another, like a specific mode of action, right? That through using our tools of microbiology, through using 16S sequencing and metagenomics, we can say, okay, these are specific modes of action that maybe this old historical culture, these magical vials that were in the freezer, maybe that's how they were working, okay? There's a bunch of cool reviews about like, now we know enough about like, I gave you the example of like the seats in the room, okay? There's different mechanisms of action of what's called CE as competitive exclusion, or CR as pollination resistance, basically the same idea. So there's nutrient or space competition, active antagonism, or inhibitory metabolites that are produced. So these are like a lot of like the modes of action of probiotics, for example. When you buy probiotics in the store, the idea is that they're generally using like one of these mechanisms of action to provide their beneficial effect. Okay, so through our metagenomics, we basically highlighted examples of each of these. So good examples. And so now maybe if we wanted to follow up on that research, so we shed light on like how this historical culture probably was working, at least these 2 ways, and gives us like future avenues if we wanted to write grants and pursue funding to pursue that. Okay. All right, so that was example 1. Example, just 2 more. So another way that we can use microbiome science to improve the sustainability of food animal production is we can identify alternative antimicrobials. I just want to give you a quick example of that. So we've all heard about bacteriophage, right? So viruses that infect specific bacteria, okay? They have all kinds of cool mechanisms, and they have like these Marslander like actual mechanisms, or they recognize cell surface proteins and they attach, and then they punch holes in the peptidoglycan, and they make these hold-ins, and then they inject all these enzymes and they blow the cell up and so on. Okay. We were doing a bunch of bacterial genome sequencing. So we're sequencing the genomes of bacteria, and a lot of these viruses, they incorporate themselves into the genome of a bacteria. It's called a prophage sequence. So hopefully you've heard about that. So we could identify in these bacterial genomes a prophage sequence. It's not hard to clone that. So you do a PCR, you pull those genes out, and then you can put those genes into like an expression vector, an E. coli or something. You can overproduce that particular protein. And so that's what my colleagues in this case did. And basically they showed that when you do that, you express this particular bacteriophage protein, set of proteins, you can selectively target the host, which in this case isn't shown here, but it's Clostridium perfringens. Clostridium perfringens is kind of one of the most common foodborne illnesses, a very common human and animal pathogen. And so, without going into great detail here, we could basically identify bacteriophage sequence that we could use as a targeted antimicrobial for Clostridium perfringens. Okay. And so the idea is that's an improvement over like these broad spectrum antibiotics that are kind of indiscriminately killing our the gut microbiome, gut microbes, many of which are beneficial, right? Okay. So we got a patent for that. My name is on here somewhere, and I thought that dollars would roll in, and I've got $0 for that. So okay, back to my government pension. Okay. All right. So the last example I want to give you here is what Gustavo he figured out was maybe we can like modulate the hydrogen economy within our chickens, and I want to show you what that means. Okay. So again, we're going back to this idea about like, can we do these fecal microbiome transplants? They can induce these big differences in phenotypes, and then can we use kind of these tools that we have to like drill down and understand like exactly why those changes were happening. Okay, so when I was still at USDA, I did some FMT, so fecal microbiome transplants. So we took gut material from adult chickens, we gave it to day of hatch chicks, and then we just let them grow and see when we saw what happened. Okay. And then Gustavo showed up at my door and I said, oh man, I've got this great data set. I need some help. Okay, so long story short, if we compare the phenotypes of birds that received this FMT, the fecal microbiome transplant, so inoculated versus uninoculated, that's basically an experimental setup. We could see that depending on whether the birds were male or female, in both cases, there were significant differences this is body weight at the end of 6 weeks. 6 weeks is the life of a broiler chicken. Okay, short and sweet. So these little guys turned from little day of hatch chicks to the full-size chickens that we buy at the grocery store in 6 weeks. Okay, so in our experiment, after 6 weeks, the birds that we gave this FMT, this fecal microbiome transplant to, they had significantly higher body weight. Okay, so that was important. And then we also noticed that they had significantly better feed conversion. So through the experiment, my colleagues and I at the University of Georgia, they measured how much feed they were giving these birds every day. And based on that, the ratio of the feed to the body weight, they calculate what's the RFI, the relative feed index. So basically for this parameter, the lower the value is, the more feed efficient those birds are. Okay. So they consume less feed to grow to the same body weight, let's say. All right. So we can see a significant difference and a significant improvement between the inoculated birds versus the uninoculated birds. So we were like, cool, we did this FMT experiment. We indeed have this like significant difference in phenotype. And now we have these magical, amazing tools that we can drill down into the microbial community and start to understand why. Okay, so Gustavo produced these two figures so we could look at 16S. So again, we're looking at like the taxonomic composition of the community. And so this color here, these are the control birds. The blue is the inoculated birds. So we could see the communities are different. So different types of bugs are there. And then this panel over here is using metagenome. So we're looking at like the whole genomic repertoire, right, of all the bugs that are there. And the same thing, we could see the inoculated versus uninoculated, different sets of genes there. Okay, so that's cool. So it does seem like this really led us to believe, right, that the phenotype that we observed is probably driven by the microbiome. The phenotype is different, the bugs are different, but we're still like, what exactly is different? Okay, so Gustavo carried on at his computer. And one of the main things we noticed was that, so this panel is, we're looking at, so you want to ask like, of the bugs that are there, which ones are over enriched in the desirable phenotype in the FMT treated birds, okay? And so one thing you noticed was that archaea, so not a bacteria, but archaea, were really highly enriched this is enriched in the FMT treated birds versus the controls, okay? So that was, we could see that, and then we could use the metagenome data, and we could say, what genes, what specific metabolic pathways were enriched in those birds too. And this was one of the main things, which is super interesting. So hydrogenases were highly enriched in the FMT bird. So this color here, this kind of salmon color, these are the inoculated birds here. Every kind of vertical thing here is an individual bird. And the gray color is the control bird. So it's like the inoculated birds are here, the control birds are here. And there's just in a nutshell, it's not biochemistry, but there's two main classes or types of hydrogenases. There's hydrogen producing, which is shown here, and hydrogen H2 consuming hydrogenases, okay? And you can see, you don't need like a PhD in statistics, you can just see with your eyeballs that the inoculated birds down here in the salmon color had way more of these H2 consuming hydrogenases compared to the control birds, okay? So cool, but what does that mean? Like, what's the, can we kind of make sense of that? Turns out that's exactly what's happening in the rumen of cows, okay? So really like basic microbial ecology of like a rumen. Okay, this is literally like a slide from a ruminant talk, a paper, okay? What's happening is the feed is coming in as these complex carbohydrates that we talked about. All these different microbial enzymes, right? These little enzymatic scissors are chopping up that feed, okay? They're oxidizing it to produce these short chain fatty acids in an anaerobic environment, okay? So things like acetate, propionate, butyrate, these are all beneficial to the host. It's like a way that we and ruminants and our chickens, that they're getting energy from their feed, okay? The process of that oxidation, H plus is acting as an electron acceptor. So it's getting reduced to H2. Okay, so you get this buildup of H2 in that ecosystem. All right. And it'll build up. And if you just remember like the kinetics of any reaction, it'll become a negative regulator. If you have too much of the product, then the reaction won't keep proceeding. Okay. And these are beneficial reactions. This is what we want to have happen. This is how the host is getting its energy. Okay. So too much H2 is going to stop that. It's going to clog up that pipeline thermodynamically. Okay. So canonically, there's kind of three main pathways. Classically, there's three main pathways by which H2 is consumed in a microbial ecosystem. There are methanogens, which are archaea, right? Acetogens, sulfate reducers, and a few other ones. Okay, but those are the main ones. All right. Methanogens happen to be their ability to use hydrogen. So methanogenesis, just the definition of the reaction, is in this case, it's combining H2 with CO2 to form CH4, which is methane. Okay, that's why cows they have all these archaeal methanogens within their rumens, and that's why cows are produced. They're burping, they're belching all this CH4 as methane, and that's undesirable from an environmental perspective because it's a really strong greenhouse gas. Okay, what Gustavo figured out in our chickens is that same thing is going on, and it's actually beneficial to the bird because it's the methanogens there are consuming all this hydrogen, and that's unclogging this pipeline thermodynamically, and it's allowing the production of these beneficial short chain fatty acids. Does that make sense? Groundbreaking new knowledge. Come on.
We're 3 minutes over. I want to keep.
Okay, perfect. All right. Okay. All right. Good. All right. So good. Yeah. Sorry. All right. Good. We get excited. All right. So this is basically, so the take home message is that the cecum of a chicken is functioning exactly like the rumen of a cow. Okay. And so now through these tools of microbial ecology, we now know that chickens are actually hindgut fermenters, and it's really the microbes that are driving their phenotypes. So that's it. And it's really thanks to Gustavo who figured all that out.
Oh, no, wait, man, it's a team effort for sure. And anyway, again, we're a couple of minutes over. I apologize. Hearing about my own research, I lost track. I didn't know that was gonna be the end, but anyway, again, as we did last time, if it's okay with our speaker, and if no one's coming in to take over this room, I think that we can open up the floor for maybe 5 to 10 minutes
Sure,
for
yeah,
questions for those of you that are able to stay. And go ahead.
Sorry, I'm just really curious about the secret vials in the freezer.
Yeah.
That had those magical unknown formula.
Yeah.
Did they make you? I don't know. sorry. Did you put it into chickens and then do 16S sequencing, or did you sequence the vials?
We just sequenced the vials directly. Yeah, yeah, because like I said, they had previously shown it to be effective in reducing pathogen colonization when they fed it to chickens.
Was there like, I don't know, maybe some suspicion that maybe once it interacts with an existing microbiome of a blank chicken, I guess, it would actually like change from the original formulation research?
Yeah, that's a good question. And I think the idea was, again, that with food animals, you have this kind of opportunity to start at the beginning of life with like kind of a blank slate. So if you provide them this beneficial community when they're young, like naive chicks, they don't have an already established community, then you can get your beneficial bugs established. What will happen to those bugs after they themselves colonize the chicken? You know, for sure, they're not going to, that community is not going to remain stable. And so like any, you know, maybe you could think of it like any medication, it kind of has a declining efficacy through time. But yeah, it's interesting questions.
I have one.
Yeah.
So you know how you said, you know, the chickens grow quicker with less feed because of the microbiome?
Yes.
But does that have like any effect on the actual meat itself, like the quality of meat?
Yeah, it's a good question. Not that I know of. I think they're just becoming more feed efficient, so they should be getting the same, you know, amino acid profile, the same. They're eating the same types of feed, just less of it as they become more efficient. So it should lead to the same meat quality and so on. But the industry is also concerned about that. There's, you know, if you stress an animal when it's harvested, it can lead to there's something called DFD, dry, something like the meat will get like undesirable characteristics. It's like,
Yeah, I think they do it with deer, right? I think deer, when
That's-
they hunt deer
right.
meat is it's scared or something. I think
yeah,
the meat or the
exactly.
stress effect.
Right, you're supposed to reduce the stress on the animal. That's a little bit different than kind of a direct microbial effect, but same idea.
Oh, no, I'm wondering, we talked about livestock, but what are the applications of microbiome studies when it comes to like crops?
Crops, so many, yeah, so you can, I started out with actually looking at, in my career, looking at nitrogen fixing like root nodules, so nitrogen fixing plants, so you can optimize those. People are like, you can hasten, you can speed up the colonization process of like rhizobia, for example, are one of the important classes of nitrogen fixing root symbionts. So you want them to colonize the roots as quickly as possible and form these nitrogen fixing nodules so the plant is able to take nitrogen up more quickly. There's all kinds of like the phyllosphere is the term for like the surface of plant leaves, okay? So you can, and there's all kind of like plant pathology. Oftentimes they're looking at like leaf pathogens that, you know, causing different blight and different spot disease and so on. So the same idea here, you could like, you know, spray on a beneficial, like a probiotic community and maybe, you know, competitively exclude, for example, some of those plant pathogens, stuff like that. And the use of antibiotics, some of those, there's some diseases like in Florida, citrus greening, I think it's called. And I think historically they would spray from like crop dusters or spraying antibiotics on these fields of citrus or whatever, however they grow them, which is a horrific thing to do environmentally, just to like reducing into emitting into the environment like massive quantities of antibiotics, right? So the idea is like, can you, instead of doing that, similar to what my approach here is, can you like have some kind of a more benign like microbiome optimization approach where you eliminate the pathogen or reduce the pathogen? Yeah.
Is phage research kind of big in this niche of, you know, agricultural microbial optimization, you know, like kind of finding
Yeah.
a therapeutic phage to deal with this instead of spraying antibiotics, you could spray it.
Yeah, good question. It should be bigger than it is, I think is the short answer. There's always been kind of a resistance to it, pun intended. People haven't been that like phage, bacteriophage in humans is like, it's just been really common like medically in like a lot of Eastern European countries. And apparently you can like buy over the counter, like, you know, salmonella phage or whatever. I don't know why it hasn't been very common and it's been kind of resistant. The research unit I was in, our research leader, he was actually kind of one of the experts and he was always trying to get his research like more widely accepted, and it seems like the industry wasn't that into it. I don't know if there's like a public perception that like viruses are bad and we don't want to be like spraying viruses on our food or food animals or something like that. But
Yeah, I had a question about, like bacteria that are antibiotic resistant.
Yeah.
So, I think you explained how you can actually transfer, bacteria microbiome to like, chickens and stuff like that. Could
Yeah.
it be translated to humans, as a way to, kind of lower the numbers? Because I recently seen that.
so you mean like a fecal microbiome transplant
Yes.
to combat specifically antimicrobial resistance? That's a good question. People like, if you think about FMT, a fecal microbiome transplant, it's kind of like medieval in a way, right? Where it's like this, it's like, bleeding someone with leeches or whatever we used to do, right? Where it's this very undefined treatment. And that's why, like I showed you, like the FDA wouldn't approve that mucosal competitive exclusion because they're like, this is some undefined goo of stuff and we don't know what it is, right? Okay. With humans, FMT has really only been a measure of last resort. So the main application of it is for people with C. diff, Clostridium difficile. And I'm not an expert in Clostridium difficile, but apparently when people get it, it's like very difficult to get rid of. And it really destroys people's quality of lives, like they can't live a normal life. So the human patients are desperate and they're like, and the doctors be like, okay, let's try this. So there's actually like some stool banks, like MIT was running one for a while, where like healthy donors you can provide a stool sample and they screen it for pathogens or whatever. And then a gastroenterologist can go to that stool bank and they can say, hey, can you, age match me with a donor to match the recipient? And then we're going to give them an FMT. And so when they do that with people that have these C. diff infections, they're actually super effective. And people like have significant resolution of symptoms. And so there's actually companies that are trying to kind of like commercialize that. Can we like, they start out with like an FMT, but then they passage it through like, like if you think of like a microbrewery with big stainless steel fermenters and stuff like that, they try to get like a stable community. So, which is really similar to the magical vials that I had, except now, again, we have the tools where they can characterize exactly what's in that. So they can say, we know there's no pathogens, we know there's no antibiotic resistant genes, et cetera. And now we kind of have this like defined, it's still a complex community, but it's still more defined. And then we can give that to patients that are really in a desperate situation. So that's really kind of the main examples of FMTs, which is maybe a little bit different question than what you asked, but yeah.
And can I do one more question? Another question? Yes, sir.
Has there been like a push in industry to do like the fecal transplants and like more livestock?
In
Or I guess
livestock?
it's the economic feasibility of doing a transplant.
Yeah,
Every single one is-
right. Again, I mean, it's like a research tool, and you can imagine if you're like, Tyson or Cargill, one of these massive companies, you're not so interested in like taking risks and like, hey, so the size of most commercial chicken flocks, I don't think I mentioned, but it's like 10 to 20,000 birds. Okay, so they have these big poultry houses. So they're not really in the business of like, hey, let's do this and see what happens, right? But they are interested, and that's kind of why I pursue this research, is like, can we understand like mechanistically exactly what happens when you get these desirable phenotypic outcomes? And then can we make like a more targeted kind of a pharmaceutical type approach? So hopefully somebody from industry will at least read our paper and they'll be like, hey, even better, they could call me, develop a collaboration. But this is the way research goes. Like you never know. You put stuff out there and then you never know exactly how it's going to go. So, but yeah, good question.
On that note, I'd like to thank our speaker again.
Thank you guys.
Thank you all for sticking around the extra 15 minutes and questions, and fantastic. Thank you. Take your seat.
That changes the hydration.